Hello students, welcome to Module 5.6.1, Introduction to Animals. This will be the features of the animal kingdom that we will be talking about in this module. Oh, and there's a big animal. Okay, so the first feature that we can mention is that animals are multicellular eukaryotes that have ingestive nutrition. It means they eat parts or entire other organisms. This is the way that humans eat. This is the way this little cartoon eats. I'm not sure what that animal is supposed to be. It looks like a bear, but it has the teeth of a rodent, and so it's probably neither. A second feature of animals is that animals have cells that do not have cell walls, so they lack cell walls. Most animals are motile. Motile is the same as mobile, so we talk about motility, same way we talk about mobility. We also include four other special features, which include symmetry, cephalization, body plan, and development. And so let's talk about those for the special features. Symmetry is how parts of a body relate to the other parts of that body. And a line of symmetry is a line that divides a body into two mirror image halves. This famous sketch actually has bilateral symmetry, and we'll see there's three types of symmetry. One possibility is for an animal's body to have no symmetry. That is, there is no single line or plane of symmetry that can divide it into two mirror image halves. An example of this would be phylum Porifera, which I know you haven't learned the phylum names yet, but you will, and Porifera is the phylum of sponges. A second possibility with regard to an animal's body is that it could have radial symmetry. Radial symmetry is when there are multiple lines or planes of symmetry that divide it into mirror image halves. An example of that would be phylum Cnidaria. This is a phylum of jellyfish. Sea anemones are found in here as well. And the advantage of this type of symmetry is that an animal with radial symmetry is equally ready to receive food to its body from any possible direction. And so it's good for sessile or attached forms, animals that are attached. Think of a sea anemone there. A third possibility, which is the most common type of symmetry, is for an animal to have bilateral symmetry. There is one line or plane of symmetry that divides the body into mirror image halves. I put here as an example Arthropoda, but I could have put here almost any of the animal phyla, as the vast majority of animals do have bilateral symmetry. And an advantage that we'll tie to one of the other special features is that animals that have bilateral symmetry are able to rapidly respond to stimuli, and we'll see why this is when we consider our next special feature, cephalization. That'll be after I show you this slide that shows the two types of symmetry, radial symmetry and bilateral symmetry, but then there are really three types, the third type being asymmetry, or not having symmetry. And you can see several sketches representing that. So a second special feature is cephalization. Cephalization is the evolutionary development of a head, and this happens by the anterior aggregation of sense organs and nervous tissue used in decision making. So if we're talking about anterior, that means toward the front. And it happens that sense organs and a brain is moved to the front because that's the part that goes through the environment first. And so I want you to consider, as an animal moves through the environment and it encounters a novel stimulus, it bumps into something, it may be crucial to that animal's survival how it responds to that novel stimulus. Is it a predator that's going to eat it? Does it need to run away? Is it food that's going to run away from the animal and therefore it needs to pounce? Or is it just a bump in the road that it needs to walk around? These, the first two anyway, are life and death decisions. Encephalization may be seen then as an adaptation for moving through the environment, and it does really provide rapid response to diverse stimuli. The distance the brain and the sense organs are apart, it actually slows response times, and so quick response times are allowed by having the sense organs in the front, where they'll first see novel stimuli, and having the brain very close to them, where it can give rapid instructions as to how to respond. And so we see that cephalization and bilateral symmetry are closely associated. For if an animal is cephalized, that means it has a head or a front end, and therefore it has a back end. If it has a front and a back, it has a right and a left. So if it has a front and a back and a right and a left, it has bilateral symmetry. And so you can see how cephalization and bilateral symmetry are closely associated. A third special feature is body plan. And body plan has a number of manifestations. We're going to talk about having a gut and having a body cavity, these kind of things, different features. So let's look first under body plan at a gut. And there are two types of gut that we're going to talk about. The gut should be seen as the food tube, or the gastrointestinal tract is another name for it, GI tract. And one possibility is to have a sac-like gut, also known as an incomplete digestive tract, where both there's a single opening through which food enters and waste exits. A second possibility is to have a tube-like gut, and this is to have a complete digestive tract in which you have a separate mouth on one end and an anus on the other end. And this actually allows specialization and regionalization. Think, for example, of your gut or the gut of a vertebrate animal. You have a mouth that has teeth that physically chews up food and add saliva for the beginning of chemical digestion, then an esophagus that goes to a stomach, and in the stomach we have an acidic environment that kills microbes and further digests the food. The food can then go to the small intestine, where chemical digestion takes place in earnest, and also absorption of nutrients, in the large intestine, where absorption of water, and then a fecal pellet is formed for defecation. And so it gives specialization all the way around. Now, another possibility under body plan is a body cavity. A body cavity is a different space than the gut. And for this, I think you should make a little diagram of sorts. There's one here on the screen, but I'm going to just draw one just to show you. And so, for example, one possibility is to have no body cavity, and so you should draw a circle. Whoops, that's not working too well. You could try drawing a circle, and inside there draw that. This would be the gut. And to have no body cavity means that this is all solid. So draw something like this and put here no cavity or no body cavity. And that's one possibility. And so all this that I've drawn with the cross-hatching, that just means that there's tissues and organs that are between the gut, which is the food tube, and the body wall. Okay? And an example of this would be phylum Platyhelminthes. So these are the flatworms, or one example, that have no body cavity. So there's a gut cavity, but then this is all solid in here. A second possibility with regard to gut is to have a false body cavity. In this case, make your little, oops, that's not what's supposed to happen. In this case, you can draw, there's the gut, and you should label that as gut. And draw that a lining on one side. So I'm going to label gut, I'm going to label lining, and then this space right here would be the false body cavity. Now this is typical of certain animals think of a nematode worm, for example. Notice here it is sliced in half with our samurai sword, and you can see that it's lined on the outside but not on the inside. And so let's look at the third possibility, is to have a true body cavity, and we'll talk about the functions of it in just a moment. You should go ahead and draw something like this. So there's our gut. It's lined on the outside and it's lined on the inside. And so this is the gut. This is a lining. And this space is a false body cavity. So you should make such drawings. False body cavity. Oh, I'm sorry. That's not a false body cavity. My mistake. It's a true body cavity. And it's lined both on the inside and the outside, and so that has a true body cavity inside there. And the cavity, it says here, is located between the gut and the body wall and has a lining on both sides. And an example would be a human being. Look, we cut this guy in half with the samurai sword. I don't know how he's going to run very well. But there's the food tube, and you can see that the other is a body cavity that's lined. And it's lined by a special tissue called mesoderm, and we'll talk about what that is in a little bit. So still talking about the body plan, another aspect of the body plan is segmentation. Segments are repeated body units, and there's three possibilities with regard to segmentation. We can have an animal that's not segmented, as a nematode is not segmented. We can have an animal that is segmented, and its segments are all similar. This would be an earthworm. And a third possibility is to have differing segments, like the segments that make up an insect. And if we look at this, this shows line drawings of all three. Here in the upper left, a nematode; in the center, a portion of an earthworm; and in the lower right, a primitive insect. So it can show you, you can see the three possibilities. So one last feature that we want to talk with regard to body plan is development. Animals undergo this kind of programmed sequence of developmental stages, and it's very diagnostic of the way they undergo the process. Now there's a whole series of terms here. I'm going to show them to you on a subsequent slide, so we're going to skip them for now, but I'll come right back to them on the next slide. But many animals have a larval stage. For example, here's a little crab larva, and in the case of the crab, it's used for dispersal. We can see down here on the bottom of the slide the definition of larva. It's a free-living, sexually immature stage of animal development that gives rise to an adult. In this case, our immature crab larva would give rise to an adult crab. Now, I think you should make a drawing of this. So draw a little circle and put zygote. And in there you can also note that the zygote is diploid, or 2N, and it will undergo division by the process of cleavage. Cleavage is rapid mitotic division without growth. And so the zygote is as large a cell as you'll have, and then it will divide into two smaller cells that have the same cytoplasmic volume, and then the two will divide into 4, et cetera, et cetera. And so this will keep going. And so that's the definition of cleavage: rapid mitotic division without growth. And eventually, this will create a solid ball of cells that is called the morula. Now, the morula will continue to divide, and because the cells move unevenly, it will create a hollow inside. The hollow is called the blastula, a hollow ball of cells. And in this case, we're showing it sectioned. And you do need to know that the space inside is called the blastocele. The blastocele is that space inside. And then through a process called gastrulation, sometimes called gastrulization, but gastrulation, a gastrula, the next stage, is formed. And this happens, think kind of poking your finger into a balloon, and when you poke it in, you can see it, and if you pulled it out, it stayed poked in like that. You would have made an opening from the outside to the inside, and that's called the blastopore, and you need to know that term right there, blastopore. And you don't have to know this fancy word, archenteron, but instead, the hole here, we're going to call it the primitive gut, which is what archenteron means, primitive gut. And so you've made an opening to the outside, you've made a new opening. I want you to know that the blastocele that was over here is still there. It's just right in here. So there, it's still there. And in the gastrula, we have the formation of the 3 primary germ layers. And the 3 primary germ layers are ectoderm, endoderm, and mesoderm, and they give rise to all the tissues, these 3 primary germ layers. And so on the, I guess I can show you on here, since some of them are labeled already, well, there's not, I can make a new drawing down here, and so if we're going to make a gastrula, we can make it something like this. And so on the outside is ectoderm, the inside is endoderm, and then we can have mesoderm form in various ways, but in this case I'm making it kind of look like Mickey Mouse or ears, and mesoderm kid form. Sorry about that, students. So then you get the idea, those 3 primary germ layers, my little scratchy drawings there. Ectoderm, though, lines the outside. It forms the skin and the associated tissues. It also forms the nervous tissues, including the brain and the sensory tissues. Now, when we say sensory tissues, it means like the retina of the eye, the organ of Corti of the ear, the parts that are made of nervous tissue. Endoderm forms the lining of the gut, and mesoderm forms muscle, bone, glands, and other connective tissue. Now, it turns out that some of the organs are actually formed from endoderm as well, but for our purposes here, this is okay. You can actually see what I'm talking about here. Ectoderm is the outside and the nervous system, and mesoderm forms many of the internal organs and muscles and connective tissue, but notice endoderm does the gut, plus it does the lungs and the liver, as well as the gut. And with that, we're going to conclude this introduction to the features of animals in our first module about the animal kingdom. And I will just remind you at this time that the animal kingdom is the last kingdom that we're going to study this semester. That's the good news, I suppose. The bad news is it's the largest kingdom and has the most species, and so it'll take us longer to study it. With that, we'll sign off.